I did my honours project for my biological science degree with the Liverpool Lung Project, which I think Professor Tim Halliwell might know them. So I look forward to some nice questions from you at the end. But basically, so I looked at, as I said, it was a non-small cell lung cancer, specifically two histologies of adenocarcinoma and squamous cell carcinoma. So basically, those have been like investigated quite a lot, but they haven't really in the research. It didn't really have that much on how gene expression changes across T stage. So T stage basically just like the tumor size and like development. So I thought I would investigate that. So I got well, my supervisor set me up with some data from the TCGA, so that's the Cancer Genome Atlas. And basically, I had to learn how to use the R programming language, and basically design scripts to pull in the data and run it as a run different scripts to like clean up the data, and then also like actually run the RNA-seq analysis. So it was looking at the level of mRNA expression across the different T stages. So I could see how the differential expression was changing as it moved along, and so I got some quite interesting results, and I'll go through some of it now. I'll try not to bore you too much, but I'll go for it anyway. And this is just sort of showing you why I sort of wanted to hit on T stage, why it's so important. This is a survival curve I plotted from using a ggplot, which is a package for R. And basically I used the clinical data to plot a survival curve for the different T stages. You got T1, T2, and T3/4. So as you can see there, if I sort of nip over to the board, you'll see like this is days from surgery, so you got 2,000 days post-surgery. Like you're seeing a massive difference between if you catch it at T1 stage and you catch it at T3/4 stage. So that's why I wanted to look at that and see, was there anything in particular in gene expression wise that was causing us, like was allowing the tumor to progress to T1 to T2 and onward to T and 3, 4? Because potentially, like, well, the idea was to see if there were any therapeutic targets or anything like that. So I'll move on to sort of how the sort of my project went. So I had data acquisition and processing. I'm not going to talk about too much about that because I think I'll probably bore you all to tears. So I used the RStat package for that, and also the RStat package for the RNA-seq analysis. And that was just basically scripts. It came whenever I ran the RNA-seq analysis, it came out with I think a p-value of 0.001, and I think about 10,000 genes came across differently expressed. So I had a bit of problem there, just too much data. So that sort of actually like led me on to the next step of the project, like as I was working on it. So I did gene co-expression analysis. We basically just looked at, was there clusters of different genes that were had similar patterns in terms of were they up-regulated a certain amount or down-regulated a certain amount, just to sort of see if there's some some sort of like superstructure DNA like changing the expression of like groups of genes. So and then on to that again, I tacked on another sort of a bit extra. This was maybe a bit I don't know. I was sort of dreaming doing this bit because it was tough, but it was again using the RStat package in Cytoscape, which is Cytoscape like allows you to make links between different like the different genes. It allows you to like form a network. I'll show you them in a second, and it'll be a lot more clear. But and that was something that I hadn't actually really seen done before, so that took for like the bulk of the project in the end. So I'll flick on and I'll show you a bit more. This is the gene co-expression analysis now. So we'll see these three sort of clusters here. We've got the big cluster A, cluster B, and cluster C. It's gone a bit blurry, but these are sort of like the different trends seen in each one. As I said, I think when I cut it down to like the most stringent p-value that I could sort of go down to, I got about 148 with it was significant across T stage, and these were displayed in here. Anything that was below three in a cluster I didn't put in, but you can see there, like in cluster A, you've got things like surfactant protein C, which obviously, like, it's potentially why I thought surfactant protein C was so like down-regulated was, well, there's two options. Either it's such a burden on the like the tumor cells to produce surfactant, because surfactant is so important in the lungs, that they maybe like try to down-regulate it so there's less of a burden to produce surfactant, or it could be that it's just the de-differentiation of the lung cells. So I wasn't too sure about that, but that's sort of like how the co-expression analysis worked. I got the different trends in cluster A, B, and C, And I'll now talk about the methylation analysis. So this is sort of how the flow worked. I had my differential expression from the RNA, and I took that and I downloaded the raw TCGA methylation data. So if you know like about methylation, you know how important it is in the genome. It's like important for switching on and off like genes, different parts of the genome, because it helps it fold up and or come apart. So I used that, and I used Cytoscape again and a package called J-active modules, and it allowed me to plot places along the genome where these gene sites were, where the methylation sites were, and you had low p-value such as this here. This would be like a low p-value ones that I displayed in red here, and when there was two of them together, they would be very significant that was affecting this gene. So that's what I went for, and this is sort of what resulted in this is the flow to get me my data, and I will show you some results that came out of this. The reason I wanted to go down the look at methylation data is, as I said, I had so many genes that were differentially regulated, so I wanted to add another factor in that would allow me to cut it down, because like when you're like basically working by yourself, like trying to troll through, I don't know, hundreds and hundreds of genes, you can't pick out the important ones. So I thought adding methylation in as another factor could like cut it down and make my work a bit simpler for me. And this is one of the results that came out. Now this is alcohol dehydrogenase 1b. And what I found out was it was actually decreased across T-stage in both histologies, both squamous and adenocarcinoma. And as you can see, it reduced down here. It was a very stringent p-value, so it should be very significant. And this is a flow diagram I made. Just I looked up the sort of the metabolic flux of all trans retinol, which is then broken down via ADH1B there, and to all trans retinaldehyde, and then another gene called RALDH1, well, the protein product of the gene, then converts it to all trans retinoic acid. What all trans retinoic acid does is it binds to retinoic acid receptors that actually regulate transcription and things like that. So if you see these arrows here, I sort of, this is, other papers found this as well, but I sort of like threw it all together, that the sort of like the tumor is sort of, it's sort of pushing the all trans retinoic acid towards degradation, because it will cause transcription regulation. So all trans retinoic acid is sort of pushing here or back up to all trans retinol, but it will be broken down by another enzyme. Because you see ADHP1B is down regulated, so there'll be less going down that way. And then even when you're down here, CYP26A is up regulated, so there's more getting degraded than there is being binding to the retinoic acid receptors. Obviously this is like sort of a theory, but other people have found it. There's a link down there if you want it. I'll pass it on to you. And then another one I had was, oh, and sorry, the main reason for this, well, this could be a potential therapeutic target, so you could maybe try to push the metabolic flux back in the way to actually bind to the retinoic acid receptors, or even a synthetic analog. So, but obviously that's a lot further for someone else to take on. And this is another one. This is these are two methylation ones that came out very similar. This is Wnt5A, which, if you like, I've done lots of cancer stuff, you'll know all about it. Like, it's important in like different like regulation of apoptosis and regulation of transcription and all sorts of things. And that was found down-regulated in adeno, but not really affected in squamous. And then CA was it was sort of came with Wnt in the J-active module, which grouped it together. So what limitations did I have? Well, batch effects, because all my data came from the TCGA, which gets it from different centers. So I had the batch effects. So I don't know, like, did they use different machines in one center, and then in a different center they use different machine, or did they have less like stringent methods there? So I had to take that into account, but that would be a lot like further statistical things put in place to work on that. And then a further would be to verify the gene findings with an external database, so possibly the Cosmic or another one of them. Cancer databases, there's plenty of them out there. And the conclusion basically I got is that there's across T-stage there's a massive differential expression of genes, and that can be attributed into methylation. And there's I think I came out in the end once methylation, I think 48 differentially regulated genes with a p-value of 0.01. So it's very significant, but there's a lot like further work to do in this sort of field to actually get any like clinical important clinical information. But that was my first attempt at it anyway, so hopefully someone will take the reins on and see what they think. But I'd like to say thanks to the Liverpool Lung Project, the Roy Castle Foundation, and also to you all for listening to me.